Welcome. This is our tutorial video on incomplete dominance Punnett squares. Mine is on paper. Yours is probably still on the computer. I think it's easier to sort of teach it when you can see what I'm doing as I'm writing it. It's easier for me to explain than just to see me typing on a computer. I don't know. I think it's easier. So that's why I'm on paper. If you also want to write it out, totally fine. Go ahead, print it out, do it along with me. Otherwise, follow along on your screen. So incomplete dominance, this is the one where we have, is our first exception to Mendel's laws and Mendel's classic genetics, pure dominance and recessiveness. So incomplete dominance, this is the one where we have two pure breeding parents, and when they mix, we get heterozygotes, and those heterozygotes are going to have a new phenotype that wasn't present before. So classic Mendelian genetics, it's either one or the other. There's only two phenotypes. With incomplete dominance, we get a new third phenotype. So looking at number one, in horses, some of the genes for hair color are incompletely dominant. Perfect. Phenotypes are as follows. I'm going to get out some colors. I love using colors here. So homozygous dominant are brown. Cool, easy peasy. Now, homozygous recessive are white. Makes sense. Usually white is a more recessive trait, but varies organism to organism. And lastly, heterozygous are yellowish tannish with white mane and tail. This new one is called a Palomino. If you've ever heard of Palomino horses, this is the coloring, yellowish tannish on the bodies, white manes and tails. So we've been given these phenotypes. Part A is asking what are the genotypes of these given phenotypes. So first up, brown. What is the genotype of those brown horses? I used blue, so I use blue again. So brown is homozygous dominant. Homo means the same. They're going to have the same capitalization. Dominant means they will be uppercase. So they don't give us letters, so we can just choose whatever. Since brown is dominant, I'm just going to choose B. So it'll be big B, big B. Homozygous, same capitalization, and dominant means a capital letter. Next up, I used orange. Homozygous recessive is white. So what does the white genotype look like? Homozygous recessive. Homo means the same. Recessive means it's going to be little or lowercase. So little b, little b. Now we have our third phenotype. Our third phenotype is this Palomino coloring. Palominos are heterozygous. Hetero means different. We're going to have one of each capitalization, one big B, one little b. Oh, I guess I should have used the colors that I used earlier. Duh. Or big B, little b. Making a mess of this. All right. Now, we did 1A, looking now onto 1B. Here. Complete the following Punnett square and ratios. All right, brown male, white female. We need to figure out their genotypes. Brown male, brown means capital B, capital B. White female, white means little b, little b. Orange, little b, little b. Easy peasy. Now we plug them into our Punnett square. Best practices state, males go across the bottom, or across the top, females go down the side. I'm just going to choose new colors because I have lots of colors. So here we go. So the male, BB, capital B, capital B. We put one allele per column there. And the female is, let's choose dark green, little b, little b. One in each row, one allele in each row. This means for each of these squares, our offspring, which are in these four squares, we're going to get one allele from each parent, resulting in two alleles total, which will tell us what our phenotype will be. All right, so the Palomino is pink. I decided to make that pink. So let's see. we're going to get a, sorry for the lag, a big B from dad, a little b from mom. That will give us a mix, a Palomino. So we're going to get a big B from dad and a little b from mom. Same thing here. We're going to get a big B from dad, a little b from mom. Moving down, a big B from dad, a little b from mom, and a big B from dad, a little b from mom there as well. All right. Number 3 of number 1. We're kind of going in and in and in. All right, what are our genotypic and phenotypic ratios? So this side will be our genotypes, this side will be our phenotypes. How many of our big B do we have in these 4 offspring? Well, looking at it, I can tell you right away. We got, let's use black. We got 0 of those. What about our heterozygotes? 1, 2, 3, 4. We have 4 that are half big, half little, are heterozygotes. So we have 4 of those. And what about our little b, little b? Don't see any of those. If you prefer percentages, you can write 100%, or you could write 4 out of 4, or 0 out of 4, and 0 out of 4. But please notice how these have their descriptions so that we know there's 100% of this heterozygote. All right, now let's take a look at our phenotypes. We know big B is brown, and there aren't any of those, so we have 0 of those, or 0 of 4. White. White is little b, little b. We don't have any of those. 0 of 4. And our Palominos are heterozygotes. 1 of each capitalization. We have 4 of those, for 100% if you prefer percentages. We did it. We did our first cross of our horses. All right, next up, we're going to cross a brown male with a Palomino female. All right, brown male, that means big B, big B. I'm going to go back and use the same colors I used for this one, which means blue, big B, big B. And our female, this time, is going to be the Palomino, this heterozygote. All right, that one was pink. That one went pink. All right, 1 of each are heterozygote. All right, yours will probably be on the same page, but I had to make my boxes bigger, so we're going to scooch a little bit. Make sure this is all on top. Too many papers. All right, there we go. This is where computers are much better than paper. So now we need to plug into our Punnett square using my same colors as I did before. Male, big B, big B. Female, heterozygous, 1 of each, 1 in each of our rows here, big B, little b. Now we got to make our offspring, our little chilies here, our little children's, little chillins. So let me get my colors out here. We're going to get a big B from dad and a big B from mom. This will give us our heterozygous, homozygous dominant. Moving over, we're going to get a big B from dad and a big B from mom. Another homozygous dominant. Now, moving on to this last row down here. Let's switch a little bit. Big B from dad, little b from mom. That will give us our heterozygote, or Palomino. And the same thing here, we get a big B from dad and a little b from mom. So we finished our Punnett square. We have our genotypes here. What does that mean for our ratios? All right, I used black last time. All right, so big B, big B. How many big B do we have? We have 2 of the 4. What about our heterozygotes? one of each. We have 2 of those heterozygotes. So 2 of those. What about our little b? Not seeing any of those. 0 out of 4 there. If you like percentages, this one is 50%. This one is 50% because, oops, because 2 out of 4 equals 50%. We figured out our genotypes. What does that mean for our phenotypes now? Okie dokie. Brown, we know, is this homozygous dominant. We got 2 of those guys. White, we know, are those little b, homozygous recessive. We don't have any of those. We found that out down here. And our Palaminos, these are our heterozygotes, big B, little b. We got 2 out of 4 of those. If you like percentages, 50% there, and 50% there, 0% there. Hey, we did it. Our second incomplete dominance there. Get rid of this one. All right, let's look at our last incomplete dominant example here with our horses. Last one, we're going to cross a Palomino male with a Palamino female. So what does this mean for our genotypes? Well, Palomino, we know, is that heterozygous, big B, little b. I chose pink for that color. And now we can plug it into our Punnett square. We did this light purple for dad and dark green for mom. Now let's look at our babies. First up, we're going to get a big B from dad and a big B from mom. Homozygous dominant. Second one over here, we get a big B from mom and a little b from dad. That gives us our heterozygote. Third square, big B from dad, little b from mom. We got swapped there compared to the last one, but we still end up with our heterozygote. And lastly, I think it's orange, we have little b from each of our parents, little b from dad, little b from mom. Little b, little b. We did it. We found all of our children. Let's focus on our ratios. So how many big B did we have? Out of the 4, we had 1. What about our heterozygotes, our Palaminos? We have 2 of those. So 2 of those 4. And our little b or white ponies here, we got 1 of those. If you like percentages, 1 out of 4 is 25%. 2 out of 4 is 50%. And 1 out of 4 is 25%. Now let's look at our phenotypes. Focus, there we go. Brown. Brown is this big B. We have 1 of those. White is our little b. We got 1 of those. And our Palaminos, our heterozygotes, we got 2 of those. If you like percentages, we got 25% there, we got 25% there, and 50% there. That's all I got for our practice and tutorial on our incomplete dominance. We were talking about horses, brown, white, and Palomino horses. I hope this was helpful.